This is the PowerPoint for the cell in health. As you know from the material from this course, knowing what happens with the cell in health enables us to move forward to looking at the cell in both health and illness. So, as we look through this, it is important to remember that the cell membrane is a lipid bilayer, and that enables the movement of nonpolar substances and also lipid-soluble substances across cell membranes. Cell membranes are also unique in that they have protein macromolecules embedded in them. These protein macromolecules serve to be cell surface antigens to help identify self from non-self, ion channels such as the sodium-potassium ATPase, which, if you remember, that's 3 sodium out for 2 potassium in, and that helps maintain the membrane potential. And then we also have specific receptors for neurotransmitters, such as a receptor for epinephrine, which would be a beta-adrenergic neurotransmitter, norepinephrine, the alpha-adrenergic neurotransmitter, and also the muscarinic receptor that responds to acetylcholine. Now, when you look at this cell membrane, you see the cell surface receptors. So one of the ones that you see is an activated protein receptor. This would be an example of what you would see with interaction with epinephrine with a beta receptor, so that a G protein would be released, triggering a second messenger such as cyclic AMP, and then resulting in all of the symptoms or physiologic responses that you would associate with the beta-adrenergic effect, such as increased heart rate, increased dorsal contraction, bronchodilation, and uterine relaxation. You also have specific receptors where it talks about receptor opening an ion channel and ions moving in, which is what you would see at nicotinic receptors, are also at GABA-aminobutyric acid receptors. So this would be, again, an example of what you would see with a beta-adrenergic receptor. Epinephrine has triggered the beta receptor. It releases adenyl cyclase, which ultimately makes cyclic AMP. It activates various protein kinases, and one of the things that epinephrine does, in addition to giving you beta-adrenergic effects, it also stimulates glycogen to break down the glucose, and it blocks glycogen synthesis, so that epinephrine functions as a counterregulatory hormone in diabetes to elevate blood sugars. Another example that you see is when epinephrine doesn't work. So then if epinephrine is not interacting with the beta receptor, you don't have those physiologic effects. So that the receptor, the beta-adrenergic receptor, is requiring the activation with epinephrine in order to give the effects. Now, the other thing that you also have to remember with the cells is that within the cell there is genetic protein. There is deoxyribonucleic acid and ribonucleic acid. Essentially, the DNA is stored in the nucleus, and it is referred to as the major genetic protein. So the nucleus, because of the actions of DNA, becomes the control center of the cell. DNA can replicate itself. It can also produce RNA, which is ribonucleic acid, and that helps regulate protein synthesis. So when we look at RNA, it's stored within the nucleolus of the cell. And RNA can then be messenger RNA that travels from the nucleolus to the ribosome and the endoplasmic reticulum to give information that will enable the formation of a protein. The ribosome is made up of ribosomal RNA, so you see the small r next to the RNA, and as it reads the M messenger RNA, it uses T transfer RNA to connect amino acids, which will ultimately form the proteins. So this slide represents what I just described. You have the double helix of DNA, and it coils around histones, and it coils and becomes condensed as chromatin within the nucleus. It will then uncoil and begin to form messenger RNA. So, for example, when it's DNA, it has adenine base, cytosine base, a guanine base, and a thymine base. However, when we're talking about ribonucleic acid, it actually has one more oxygen than deoxyribonucleic acid. And because of that, instead of thymine, the base is now uracil. So that when we look at the various bases, if DNA is going to make RNA, what would be the adenine nucleotide would combine with thymine, the cytosine would combine with guanine. But when DNA makes RNA, adenine combines with uracil, and cytosine still binds with guanine. This is kind of important in understanding how proteins are made, because if there is a mistake in the DNA sequence of these nucleotides, we now can get an alteration in the formation of a new protein. And we actually see this with sickle cell anemia, where there is a DNA miscode, which translates to an RNA miscode, and a base substitution, an amino acid substitution, which ultimately gives us sickle cell anemia rather than normal hemoglobin. So here we have a DNA double strand, and it gives you the information for the DNA strand, but when it breaks apart and begins to make RNA, you see that there's a 3-base codon, or 3 bases in a row is referred to as a codon. So each of those 3-base codons relate to a different amino acid, so that there is a code that geneticists can look at that identifies if I have a specific base sequence on DNA, I will get specific amino acids within a specific order. So AUG relates to methionine, GGU, glycine, UGU, cytosine. Okay, and you can see UGU corresponded with AGC, or ACG, ACA. Yeah, I've got them looking at the right. Got to look at the right sequence. ACA. If you look at the lighter color, translated to UGU for the amino acid sequence. So, moving on to the next slide, we see that in the nucleus, DNA unseparates or uncoils, and this makes a new RNA, which eventually becomes the messenger RNA. The messenger RNA will travel along the endoplasmic reticulum to the ribosome, which is made up of ribosomal RNA, and then protein synthesis occurs using transfer RNA. All the little amino acids connect together, forming a specific protein. So that the other thing that is really amazing about DNA is that it replicates itself, so that I can have a parent DNA split apart to become 2 daughter DNAs, and you'll notice that they match up, so that each one is a perfect copy. and that enables genetic information to be transferred cell to cell. And eventually, this genetic information gets transferred from the parent to their offspring through the formation of an ovum or a sperm cell. So that when DNA replication occurs, there is a replication bubble where there's a DNA polymerase that kind of opens up and starts reading things so that we get an exact replica of the DNA. When we get to the ribosome has to read the process and put it all together. Now you'll notice that there is ATP, high energy phosphate, that is also involved in this process. This is just explaining again how strands of DNA can be made, looking at when it is open. Now this also is more depth of content than what would be on the pathophysiology exam for the Cell and Health. But what this is doing is giving you a picture of the process so that when they talk about DNA being able to give an exact copy of itself because of base pairing between the amino acid bases, this gives you an explanation of what you would see. Now, protein synthesis, as I mentioned before, DNA transcribes messenger RNA. Messenger RNA travels along the endoplasmic reticulum to the ribosome. The ribosome is comprised of ribosomal RNA. The ribosome reads the messenger RNA, uses transfer RNA to make the new protein. Once the protein is formed, it moves from the endoplasmic reticulum to the Golgi apparatus. So DNA, so again, this is this process where you'll notice here that instead of T and A together, you would the adenine combine make it with uracil, adenine with uracil to make the RNA. You now have the RNA polymerase in the process of making the RNA. You'll see the high energy molecules for the triphosphates, which are part of the process. So now I have a process similar to what you saw before, but this time I'm making a messenger RNA. Now, one of the things that happens is that you can get inclusion of abnormal codons or exclusion of the normal codons. And some of that accounts for the genetic miscoding that has occurred over time, or the formation of what we call founder mutations, such as what you see with hemophilia, which is descended from Queen Victoria. This is an example of an RNA loop that is coming together to make the protein. And at this point, I will stop, and you'll get the second half of this in health.